Hello, Dr. Singh.
Good afternoon, sir.
Good afternoon. So you are now refreshed?
Yeah, I just came back to home because because there will be many deliberations, so I thought that I will take from home, and now Dr. Subudhi is with us, and
Oh, that's very good.
great to
That's
see
very
Dr.
good.
Subudhi after a long time.
Right. Right.
Very good morning. I think it must be very early in the morning there.
Yeah, Dr. Singh, how are you?
Very good. Nice to see you
Good
after a
morning.
long time.
Yeah, good evening there. Thank you.
So maybe Dr. Das, you can take over, but probably we can still wait because there are only few people, maybe few minutes, and then you can start things.
So it will start at 7:30, no?
Yes. Actually,
Dr. Das.
Dr. Das, please. Yeah.
Yes. We are supposed to be by 7:40. Now we are little ahead of time. But anyway, so it is now in India, it is around 1740 hours. And what about your place in Louisiana?
It's 7:06.
7:06 morning.
Yeah, just I woke up.
So very good morning to you then.
Yeah, good evening to you. Good afternoon.
Very good. Because now I find that the people are coming from states. They are from Louisiana, then Dallas, Texas. So there will be quite a change of time schedule.
Yeah.
Different time zone. Right.
Yeah, 6:40 was very earlier.
Yeah, it is very early. How is weather there?
Yeah, it's raining little bit, drizzling.
And a little bit cold?
Yeah, not cold. It's hot now.
Oh, now in the paper we have seen that there is some snow blistering somewhere in North Dakota or other places.
Yeah, northern areas there is snow.
That's it.
Yeah, but we have actually, there is forecast there will be rain for next 2-3 days, but it is very hot, around 8 Fahrenheit. But it's like hot like India.
I know. Yeah.
In the Texas, you can see hot, you can see rain, and you can see snow on the same day.
Right. And
no, this time we got also a little bit of snow in extreme south.
Wow, you are lucky.
Yeah, this winter storm came, I think 1 month ago. It was very bad.
Oh. So how is life there? It is smooth? Or now it is?
Yeah, it's because of coronavirus. It is not yet normal.
Still now
classes are like hybrid classes, and many of them are online classes.
That's great.
I think it will normalize by fall season.
That's right.
Yeah, because vaccination is now going on. A lot of students are now taking. Hopefully it will be better soon.
But somebody was telling that by month of May, America is supposed to vaccinate everybody.
That's what our president, Joe Biden, they will have vaccine actually for everybody, but there are vaccine registers are there. They don't like to take vaccine.
Still there is apprehension. Yeah, that's right. Still there is apprehension.
Yeah, there are 20-25%, 28% people, they don't like.
That's a problem. That's a problem.
Mhm. And most of them are actually this Donald Trump.
That's
That
why
means,
yesterday there was a they had a call that Donald Trump please tell the people to have the vaccine. So they had the call on that.
Yeah, that's what everybody is requesting him because he has a big following.
Big follower, yeah.
Yeah, he can tell them to take vaccine. Yeah.
that's a point. That's
And
a
not only
point.
vaccine, also mask.
Mm-hmm.
That's right.
Mask. That is not. But in university, actually, we are going to building without mask. You cannot enter. Everybody is using mask. Most of
And
them.
that's it. That's it. Yeah. That becomes a mandatory or not
Yes.
really?
Yes, our governor, because it is a Republican state, but our
Acha,
governor.
acha. But he insisted that way. That's
Yeah,
very
he
good.
has made it mandatory.
That's it. Very good. Good.
So, Dr.
Yes.
Singh, you are in Dubai now, no?
Yes, doctor. I'm now in International Center for Biosaline Agriculture.
Yeah, I know that, but where is that?
Yeah. So I joined just in the end of 2018.
Okay.
So now I'm working on the different crops and rather taking care for the whole program leader for the crop diversification and genetics section.
Mm-hmm.
And
Yes.
we are working mostly on the quinoa or other things which are something like you can say the facultative helophyte, and that's also for food.
Mm-hmm.
Not only for food, but there is a good biomass even for the feed also, and it is better, particularly under the very high salinity. Its threshold salinity tolerance is very, very high than the rice, and you will be surprised that our groundwater salinity is 26 decisiemon. It's around 60% of the seawater salinity.
Oh.
So it's a very challenging conditions, but we are taking care of the things and looking forward for more things to come. And it's not only food, there is a feed security also in this region because of the politically volatile reason. If somebody can close the border, particularly for feed, you can't buy the feed in the supermarket. So there should be not only food security, feed security because of livestock also. Welcome, Eba.
Yes. Hello. How are you?
Hello.
Hi. Hi, Dr.
Hello.
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 So we are getting people around now. Right. We've got. And okay.
Yes, Dr. Professor Das, I'm getting my old friends on the rice.
Very good.
Old rice friends, rather, I will say.
That means you are pretty excited right now.
Correct.
Right. Very good.
So,
Very good.
Dr. Singh, how do you like working in Dubai? It's in Dubai, right?
Yeah, it's in the Dubai. Sometimes people ask, agriculture in Dubai, is it possible?
Nowadays, everything possible everywhere.
Yeah. Because they want to do whatever or what can ever be done here. And they say that what you need, just let me know. We wanted to do that. So sky is the limit nowadays.
Okay.
Maybe still there are time, so maybe just when 5 minutes are remaining, Professor Das can, I think, start something to give the brief to the participants, and then we can start.
That's fine. Now this is since morning in Indian time, we are busy with conducting a lot of lectures and very interesting lectures delivered since
correct.
morning.
In the morning there was very good lectures, and
right.
also our reporter and Dr. Nikhil just tell about that Dr. Veena.
Now we are joined, sir.
Okay. Reporter here is there?
Yes, sir. Yes, sir. He is there.
Yeah, reporter is there,
Nitish.
Dr. Nitish. Is Veena is coming in this session?
I don't know, sir, but it's like I'm ready with her presentation. If she will come in, I will be like presenting for her.
Okay, okay.
I will just slide share for her.
Because, and what about the Swami? Is
No,
coming?
sir. No, sir.
No. Okay, okay, okay. Then it's all right. Then we have got only.
I think we can start from Dr. Subudhi, sir, first.
Okay, okay. So, Professor Prashant Kumar Subudhi, it will be our first talk by him in this particular session. And he's from Louisiana State University, Agriculture Center, Louisiana, USA. And his title of the paper, Challenges, Opportunities in Designing Salt-Tolerant Rice, Development of Climate Resilient Rice, and Molecular Elucidation of Salt Tolerance Mechanisms. And in fact, he will present the progress so far in the center. So we are looking forward to listen to him. And you know that he is a very distinguished personality. He is there pretty long time and working this challenging subject, the salt-tolerant rice under climate change. So naturally, climate resilient rice and their molecular elucidation, that is very important subject. And he is going to describe the salt tolerance mechanisms. So, Professor Prashant Kumar Subudhi, I would invite you to deliver your talk, and hopefully everybody will appreciate the understanding, more of understanding of salt tolerant mechanisms. Dr. Professor Prashant Kumar Subudhi.
Okay, thank you, Dr. Das. Let me first share my screen. Can you enable the screen sharing?
Yes, please allow him to become co-host so that he can share the screen. You are mute, Dr. Nitish. Yeah.
Now you can start, sir.
Okay. Are you able to see?
Yes,
Sir.
very well.
Okay, thank you, Dr. and all the organizers for the invitation for this challenging time, the conference, very mega conference on coastal agriculture for food and income security. I would like to thank the organizers, sir, for the invitation and opportunity for our work on for sharing the salt tolerance breeding in rice. You may say, well, why we are doing work on salinity in US? Actually, I am from a state which is very coastal. It's in the coastal area, just bordering Gulf of Mexico. It's not a very big problem, but gradually the salt water intrusion is taking place, and a lot of groundwater is becoming saline now. Slowly it is saving because the aquifer is now depleting, and in Louisiana, because most of the farmers, they are using this aquifer water, groundwater for rice irrigation. And let me, okay. So why salinity is so important? Because why salinity is so important? Because we know that the fresh water is going to be less and less in future. And it is only 2.5% of the Earth's water is only fresh water. And 97.5% is saline water. That's why if you can devise ways or develop some crop varieties which can survive and yield well under saline irrigation. It can help reduce actually many of the water related conflicts in and around the world in different countries, different states. And we are seeing in India and other between different countries also, there is already fighting and for the fresh water. So to give a view of what is the magnitude of salinity in the world, you can see here around 955 million hectares of total land area, almost 7% of the total land area is affected by salinity. And in irrigation area, around 20% of the irrigated area. And particularly in South and Southeast Asia, you can see around more than 110 million hectares are affected by salinity. So let's discuss about how rice fares compared to the other crops. Here you can see the threshold of different field crops, starting from corn, sugarcane, rice, soybean, wheat, cotton. These are all major crops. And you can see here rice is better than corn and sugarcane. But and for every 1 decicime or 1 EC increase in salinity, there is 12% reduction in yield. And beyond the 3.0 decicime or EC, there is you can get actually huge reduction in yield if it increases like 7 or 8 or 10 decicime. Almost plants can die. So rice is not uniform in resisting salinity in all the stages. There are certain stages which are very sensitive, and among those stages are the seedling stage. You can see here that is most sensitive, and the flowering stage is also very highly sensitive. And in seedling stage, if you you may lose the whole crop if there is the death of all these seedlings after the rice emerges from the soil. There is huge loss. Whereas in case of reproductive stage, when the plants are flowering and there is suppose silent stress, then this can also reduce yield drastically because of sterility. What are the mechanisms of salt stress? There are 3 important mechanisms in case of salt tolerance. Number 1 is many people are actually focusing on ion exclusion because the plants should we should incorporate the traits so that it can exclude sodium chloride from uptake. That is many people are working on. And also the other one is osmotic tolerance. You will see actually in this case a lot of work on transgenic. Many of the genes have been used for doing the transgenic, creating transgenic, but I don't think there is much improvement in that. And another mechanism is tissue tolerance. Even if the salt salinity is higher, there is a varietal difference in tolerance because of compartmentation in the leaf tissues. Besides that, there are some other factors or traits like responsive stomata, because the stomata should close when there is stress, or the efficient reaction ROS system. Those are also contributing to this salt tolerance. Like any other trait or breeding for any other crops, we need actually the sources of tolerance. We should have varieties or the donors for tolerance to breed a salt tolerant varieties. We should also understand the genetics or the mechanisms of salt tolerance. And there should be a good screening methodology. And if ultimately we can find some markers for efficient transfer or tolerance, that will be helpful for the breeding program. So there is huge variation for saline tolerance in the world germplasm. There are two main subspecies of rice, which is Japonica and Indica varieties. And there has been several studies that Indica varieties are better than Japonica. And many of these donors are actually Indica varieties. In US, this actually uniformly, most of the US varieties are susceptible to salt stress. Because as you know, it is not native to US, and all the varieties which have been bred in US, they have been, you can trace it to only few 20-25 varieties. So that's why there is a huge, very narrow genetic base, and there is absolutely no tolerance to salinity. And most of the US varieties belong to Japonica group. So before I show some results, you can see here what are the challenges for saline tolerance. That is what actually I faced when I did this, started this project. I faced these problems because I use mostly the Indica donors, and most of the Indica donors are having a lot of undesirable traits. Like photosensitivity and ONS, red pericarp, saturnine, like Pokali, Nonnabucra, all these varieties which are commonly used as donors, they are having these characteristics. And the most important obstacle I faced, because when I made the cross between Indica and Japonica, when I crossed those varieties, the donors, salt-tolerant donors to our US cultivars, there absolutely there is no fertility at all. They become completely sterile. So that was a major challenge to us. And regarding the screening protocols, we use the same protocol which is used, developed by IRRI, the screening protocols in hydroponics, and we go up to 12 deciciemen or 2 EC for screening purpose. But lately, for the last several years, so when the breeding population developed and we have so many, like thousands of lines, it was completely impossible to screen those in hydroponics. That's why we started screening in sand culture. And that gave actually very good results, almost correlated with the hydroponics. But in this case, since the salt stress is not immediately sensed by the plants like hydroponics, because it's in sand culture, it takes some time to show some symptoms. We used up to 18 EC for screening purpose, and then scored this salt injury and other traits. And this is the standard selection salt injury symptoms. We evaluated in each individual plant-wise, variety-wise, that in a 1 to 9 scale, 1 being the best and 9 being almost dead. So before we started the project, one of my students, she screened all the donor varieties we got from IRRI, and as well as a lot of varieties which are in the US, bred in southern US, rice growing regions, Louisiana, Mississippi, Arkansas, those are the major, and Texas. And you can see clearly here that these blue varieties, blue lines, these are mostly salt sensitive varieties. Invariably, all of the US cultivars are susceptible to, there are little amount of tolerance. You can see here R609, LAH10. This is actually a hybrid, but it has some blood from the Indica lines. That's why it's showing some tolerance here. Otherwise, most of them are susceptible. And on the contrary, you can see here FL478, Hasawi, and Pokali, those are some of the varieties we used. This is showed actually tolerance, consistent tolerance to salinity. Now I'll discuss a little bit of genetics, because here actually most of my slides later on will be actually on breeding purpose, but a couple of slides will show in genetics aspect. We used in a Bengal Pokali population. Bengal is a US cultivar, and Pokali is a well-known donor. And you can see here the clear-cut difference with respect to sodium uptake, potassium uptake. In case of Pokali is more, whereas salt injury score also, there is clear difference between both Bengal and Pokali. And also sodium potassium ratio, you can see it. And we did in a high resolution map with about 10,000 SNPs generated by genotyping by sequencing. We mapped those traits. You can see here majority of traits are actually QTLs controlling those traits are minor in nature, with a few exceptions like potassium concentration, QK 1.11, that is around 16%. This is the region where the Salton locus is located. And there are other like cis, also is 5.1b in 5th chromosome, number 5 around 13.5. And invariably in most of the cases, actually the Pokali donor segments are responsible for the trait improvement. And because of that also, because of high resolution of the map, we could find a lot of genes in that regions. We narrowed down because it was pinpointed to very small segments of the chromosome. One interesting observation from this is regarding the Salton locus. And you might have seen, because most of you are actually working on salinity and rice, and the Salton is well-known complex locus used by people, researchers, for improving salt tolerance. But we found an interesting observation. I told you that QK 1.11 is somewhere in that region, Salton region, but we found that this is actually little bit below the HKT 1.5, that locus. And we found also that the tolerant varieties and sensitive varieties, even if there is no, that Salton locus is not intergressed, but still it is having a tolerance. That means it is not just the Salton locus which impart tolerance, but still then high degree of salt tolerance can be accomplished even without the Salton locus. So that is the essence of this slide. And then we try to improve the US cultivars for salt tolerance. We started actually making backcross populations, because as I told you before that it becomes sterile when it is F1 is highly sterile. And that's why we made backcross in several generations. That was the objective, to restore fertility. And this is one in Nonavokra in Jupiter background. You can see we could in sand culture, you can see clear difference between tolerant lines and moderate tolerant lines and susceptible lines. And in this backcross breeding program, we after the each backcross generations, we selected for the desirable traits, particularly fertility and non-shattering type and non-photosensitive. These are, we selected a very stringent selection we applied in the backcross generation. And we evaluated around 600 lines and highly tolerant types, around 16 progenies we found to be very highly tolerant. So this has done, we did it in several background, 2-3 varietal background, and we have developed some lines which are highly tolerant. This is one snapshot of how these tolerant lines which develop through this backcrossing program. You can see here, Jupiter, this is the US cultivar, its CC is 8.7, whereas in case of some of these lines, this is sodium potassium ratio is very small. This is one of the lines actually we are is very promising for release. And you can see here the performance, and many of these lines which we develop, salt tolerant lines, they are having salt injury score of 3.0, like the other, like the donors. So these are with the CHTC is from TCCP, that is one of the donors we used, and these are all with the Jupiter background, but Nunavut is a donor. And then I will spend a little time on why we need a pyramiding for improving tolerance. And based on the work done by my student, we ranked those genotypes, all the lines which are evaluated for salt tolerance. You can see here, even the Focali is not great with respect to rank not high in all the traits. So that indicates that the trait, all the traits, desirable traits, are not concentrated in each variety, each donor variety. That's why we need pyramiding, so that traits contributing to salt tolerance can be transferred or combined. And you can see here, Focali, his CIS score is high, but Focali, in case of sodium potassium ratio, in our work, it was around 10th rank. And some of them are here and there. Some are better, some are not. So we developed a scheme to pyramid these different traits from different donors. And you can see here that this is, I call it as mini magic, because the magic populations, multi-parental advanced generation intercross, that is the Erie people gave those concepts. Instead of that, I used, because it is very difficult to handle so many donors because of sterility. So that's why we use 2 donors and 1 recurrent parent, or you can use also 3 donors. In some crosses, we did also 3 donors, and F1, and cross the F1s, and the back cross done, and a couple of generations of back crosses done. And we discard the undesirable traits and develop the intervention lines with donor segments from 2 different donors. And this is what different crosses we made actually, like Jupiter and background, but Hassa we used, FL478, TCCP we used. In one of the crosses we used Dular as a combined the drought tolerance. In some crosses, Gong Bio is one variety which is a South Korean variety. It's also tolerant, good tolerance. And you can see here, we could find actually some of them are very highly tolerant lines from these populations. And you can see here, there is a clear segregation for tolerance and susceptibility in our greenhouse screening. And this is, you can see here, a lot of these middle lines, tolerant lines, and the parent is the recurrent parent, P1, P2 are recurrent parent, and the donor is, you can see here, very tall plants. Whereas we selected the dwarf types with salt tolerance. And most of the lines which we developed, they are all dwarf and high. And at the end, we see their performance in the field. And these are one line with very dwarf plant stature and maturity same as the recurrent parent, and good fertility, and there is no other undesirable traits. And from those selected lines, we did some yield trials in 2018 and 2019, This is one of the lines in 100. You can see here it compared to the Jupiter, the recurrent parental background, it topped actually compared to all the, I have data for all the medium grain varieties, this line was above all the lines, but that is having high degree of tolerance to salinity. Similarly, this is a senior background, that is a US cultivar background senior from Nona Bokra. It gave also good yield. And this is one, it's CL Clearfield rice background with Pokali. This gave also high yield in a mini trial. And these are all these lines we selected based on a lot of markers also. Those are the breeders they are using in rice station, like some of the traits like aroma, height gene, waxy gene, PI genes. These are also used for screening purpose. Many of these genes, desirable genes, are also present in those integration lines. And this is another year, 2019 trial. You can see here compared to Jupiter. As I told you in the last slide, JN100 also showed best performance compared to other lines and also compared to recurrent parent. So there are a few take-home messages from this, because salt tolerance is a very complex trait. And we usually, the breeders, we don't use it back crossbreeding for complex traits. So that is what we studied that it's a major gene transfer, usually back crossbreeding is done. But in this case, actually, we have successfully used the back crossbreeding and a modified multi-parental advanced generation back approaches to improve both salt tolerance and yield. And I didn't show much of our QTL mapping using the interrogation lines, but we have several papers published using those interrogation lines and simultaneous identification of QTLs for desirable traits, as well as the elite lines also can be could be accomplished using those interrogation approaches. And lastly, the pyramiding of QTLs or traits contributing towards salt tolerance is very much needed if you want to achieve higher level of tolerance. So what is path forward? So ultimately, my message for salt tolerance breeding is pyramiding, and pyramiding. You have to have pyramiding of traits or genes or QTLs, different ways you can do it for improving salt tolerance. One way is seedling stage tolerance and reproductive stage tolerance. I didn't show any results of reproductive stage tolerance because very little work has been done on reproductive stage tolerance. But we have evaluated some lines which are having also both combined seedling and reproductive stage tolerance. And you can use stacking QTLs for multiple traits, or that can be used by IELs we developed from multiple donors. So that's also some of this ongoing work in my lab. And these multiple parents is a wonderful way of combining the traits into a single variety using different parents. And then ultimately we need to have to discover the superior alleles for QTLs associated with salt tolerance so that we can use marker-assisted selection for improving tolerance. And then last point, I will say that most of the salt tolerance has been ion exclusion or tissue tolerance. Most of the work has concentrated. But since plants are usually exposed to low level of salinity throughout the growing season, so level of salinity starts with low level and then gradually builds up towards the end of the growing season. That's, I think, the genes for osmotic tolerance should be given also emphasis. Combine several osmotic tolerance genes which can give or improve tolerance in the long run, along with the iron exclusion and other traits. So with that, I would like to thank all my crew, the students and research associates, and the collaborators, Dr. Linscomb, he is the breeder, Adam Hose, also breeder, Dr. Jain from JNU, he has done some work in our salt tolerance, we have published on sequencing work, and Dr. Singh and Glenn for supplying us with the donor lines. And I would like to thank also the USDA for funding this project and also Louisiana Rice Research Board. With that, I would like to thank you for your patience and listening. And if you have any questions, I can answer or we can discuss. Thank you.
Thank you very much. Very systematic, to the point, and very lucid presentations. Thank you, Dr. Professor Subodhe, for your nice presentations. Now, if I can understand whatever you have suggested, that is actually your main emphasis on understanding the genetic mechanism of it. And as you have suggested that it is a complex trait. And from that angle, you try to find out QTL mapping and then QTL, different QTLs identifications. And particularly you have shown in the chromosome number, where is that cluster of genes present there. Now I was wondering, first thing, that you have suggested the two level of tolerance, salt tolerance. One is at vegetative stage, seedling stage, another is a reproductive stage. Now, did you compare the QTL mapping of both stages? Do they have the difference in number of QTLs, or do they have any new order? And what I was wondering, that it may be possible that same gene systems are present, but during the developmental stages, they are being expressed, some are being expressed, some are not being expressed. I do not know, because you are trying to explain all these things. And what is your experience about that point of view? And second point about the pyramiding, that is, you have suggested multiple donors should be there. And then, of course, the backward generation for the conventional way of doing things to restore the fertility. Now, you have suggested that in your advanced lines, as compared to Jupiter, which is sensitive, you have seen quite a bit of improvement. If I can remember in your slide, some in Jupiter, it was somewhere 700 something. And in case of highest lines, it is 1000 something. So you have got some progress. And what is the percentage of improvement did you get? And did you find that that improvement stands in different locations trial?
Okay, the first one is comparison of the seedling stage and reproductive stage tolerance. Okay, in that context, we have done actually most of the QTL mapping in several populations, both from Pokali and Nonabokra, the seedling stage tolerance, QTL. And, but we have not done yet reproductive stage QTL mapping. We have got some results, but I can infer from our observations, because my objective was to find a plant which is tolerant at both stages. So those actually JN100, I saw that one of the lines was JN100. You can see here the JN100 this is one of the lines which is highly tolerant to salt stress, and it's also high yielding. But when we evaluated it for reproductive stage tolerance, this is not tolerant to at the reproductive stage. So what it gives information, what we can conclude is that probably the salt tolerance at seedling stage is different. Mechanism is different from the reproductive stage tolerance.
Actually, if
Yes, Dr. Singh.
yeah, there is some background noise. I think somebody. Please mute your mic in case if you are not talking. Somebody is there. I will request host to mute the mic in case. Some background noise. Do you also hear the background noise?
Yes, sir. Yes, sir.
Yeah,
Is having that.
The host, please.
Let me see, sir, who is there.
Host, please mute the mic. Who is? Yeah. Okay. So you know that I worked a lot on that reproductive stage, and then there is a clear cut that when you are talking about the QTL, you won't find the saltol when you are looking for the QTL for the reproductive stage. So these are completely different. That's true, and we have some papers on that. And the another thing is that particularly, I would like to tell, like rather know from you, Dr. Subudhi, that when we are talking about the Pokali, if you, because you made the like a sequence of the different varieties and what are the prime or predominant traits which are making them tolerant. So Pokali, you put the SIS, seedling injury score, but seedling injury score is also a manifestation. So I think I will request you to go ahead on that and then find out why this, because this manifestation is because of what trait. That would be like rather more informative on that. Like when we are talking about the seedling injury score, why it is having the best. Is it because of some other traits or maybe some hidden trait on that?
Yeah, I agree with that because seedling is like an overall performance of the traits. That's why we always, most of the people actually use it as a screening that based on that we select actually the tolerant ones.
Yeah,
So
correct. Because we are taking that as a overall, and when we are taking this as a overall trait, it means it is because of something. Why I am worried, because the Pokali did not appear anywhere in the other traits.
Yeah,
so
that
Pokali,
is
that's
interesting.
an intriguing thing from my my side. Thank you very much from my side.
Yeah. You see, I see actually in. Oh, sorry.
Sir? So there is one question in question answer session. It's a kind of very interesting question. Dr. Abhishek Majumdar has asked this. Sir, in some articles, we see SES score under salinity in decimals. That is like 2.5 or something like that. But ED SES system has given scores like 13579 that we are giving generally, that we are using odd numbers. How can we be able to estimate decimal level SES scoring of genotypes or lines in a population under salinity stress screening experiment? I hope like anybody can answer this question. Actually, he's asking that SES score under.
Yeah. Actually, Dr. Subudhi can answer. Otherwise, I can also answer. But let Dr. Subudhi answer over that. When we are taking the average.
Your question is SIS scoring in population or what?
No, sir. SCS scoring in terms of scoring of genotypes, we used to give a kind of numbering that 1, 2, 1, 3, 5, 7, 9. That SCS scoring we are giving. But he's asking that in some of the articles, it is given in like decimals. So how come it is coming in decimals? Because we used to give a kind of a score to that one. So he's asking that.
Okay. So that if it is decimals is there, actually it may be average.
It's an average.
Yeah,
Yes.
it is when we are taking like a multiple observation and then we
Yeah.
take the average, then it becomes like a decimal. Yeah.
Decimal 3.3, 3.5, like that.
So basically that is from average data, I think.
Yes. Otherwise, we score it individual plant wise. When we are screening, we do it to give the score of 1, 2, 3, 5, up to 9.
Yes.
But when you over several plants, it may sometimes it may be 4.5, sometimes it's 3.3, like that. Because of that, it is decimal.
And sir, I have my one personal question to you. Actually, you have mentioned in your slide something like modified magic. What is that?
Yeah.
That is number one question. And second question is, and so second question is that you has mentioned that we have to go for like pyramiding of QTLs and something like that. So nowadays when we are having that much of kind of like genomics assisted breeding that we are going on, so why not we have discussed about like haplotype breeding. I hope we are moving in that direction nowadays, because ERI peoples, they are doing that one, and but not in kind of celebrity assisted breeding, but we can also do that one. So I hope.
Yeah. What was your first question?
Actually, my first question was, sir, what is the modified magic that
Oh,
you
modified. Okay.
Yes,
Actually, the magic population, that is what ERI published the first paper, Hailing and his group, the multi-parental advanced generation intercross. In that case, actually, they used 8 different
parents. Yes.
lines, made the crosses and different, and then go forward with the crossing, and then develop these inbred lines at the end after selfing several generations. What is modified actually, why I am saying this, because instead of 8 lines, I used only 4 lines. But out of that also, because I wanted to do back crossing, because otherwise you cannot restore the fertility in the breeding lines developed from Pokali or Nona Bokra. Because those are when you make the crosses, F1 is completely sterile. So they used mostly actually high yielding lines. So there will be a lot of segregation for undesirable traits. So that is also another reason why I used back cross breeding and using 3 to 4 parents at a time. And instead of their population development, they actually cross it. F1s, then cross it, double cross, then double crosses, again cross. So compared to that, in my case, I used back crossing. I made the first cross, made the double cross, and after that, I crossed it back to the recurrent parent couple of generations to restore fertility. So that way it is different. That's why I'm calling it as mini magic. Instead of 8 lines, I used only 3 to 4 lines. And the other question, what you asked about genomic selection or haplotype breeding.
Yes, sir. It's like, sir, I can say about haplotype assisted selection. It's like a smaller version of that one. In that we can also go for that one, because nowadays I don't know that anybody started with that or not, because in other traits, in rice they are going on, but not in cellulity kind of thing. That has not been, I have not heard about that.
Yeah, it is a really complex trait, and people are talking about genomic selection. I do not think in public sector breeding, genomic selection, anybody will be doing that, because it involves a lot of work, and you have to have skills and also manpower to do that, and it is also expensive. Because now in US also, none of the public sector breedings are doing program, they are doing genomic selection. Only I heard in case of wheat, they have a consortium. So that is why they can, in a training population, they are sequencing it. That is already a lab involved in genotyping all these lines, training populations, and then calculate the GAVVs, genomic assisted, the enabled breeding values, and then they send those values to the breeders to make the selections. So that is not happening, I don't think, in case of public sector breeding. Private sector, yes, they are doing that. But there is also criticism for genomic assisted genomic selection, is that because it is not, there are a lot of variables are there, because we borrowed it from mostly animal geneticists. And mostly the milk production capacity or other things they used, because in that case they are in a very controlled condition. That's why they made a huge improvement through genomic selection. Whereas in case of plants, that is a lot of variables, environmental variables are there. Whether it will work, there is criticism. There are several papers also, review paper we can find, where there is criticism that in plants, it may not be that effective.
Okay, I think we can go
Professor
ahead.
Subodhi, may I put forward a question to you? This is Lahiri Majumdar. You mentioned that you need to pyramid the characters in from different germplasm. Suppose, have you been thinking of taking those germplasms which have been raised through the transgenics? In transgenics, there are specific genes that have been incorporated, and some salt tolerance have been shown by through a number of genes. We know of several genes which have been successfully incorporated into rice, sensitive IR64, IR29 rice, indica variety mostly, which given no compromise in yield, but also salt tolerance up to level quite high in comparison to the Pokali or Nonabokra. Have you ever thought of using those transgenic lines, use them as germplasm in your breeding purpose? Do you have any plans or any idea about it?
The answer is no, because in US actually I stopped. I did some transgenic work in the past, but I stopped working on the transgenic because in this case, in US rice growers, they are not going to go for this transgenic. That is the main reason why, because I don't want my breeding lines or any of my lines to be contaminated with any transgenes.
Okay, now you can try that with the gene modification, gene editing procedures.
Yeah, gene editing, but still, this is a good, actually, academically it may be good, but thing is that if the farmers don't like it, there is no way to do it.
The results of the experiments go to the farmers only after academicians start the work, scientists do the work, and actually they are translated into the farm's field. Isn't that true?
Yeah, that is true, but in here in the US, actually, we can't grow the transgenic. That's why I stopped working on transgenic, because the farmers, they don't want it in rice.
Louisiana is a state which will not debar you only. Yourself and the other persons are also there, Erie persons, right?
No, they will not debar, but I will not get funding to do the work.
Okay, it's a very debatable questions. Let us put it here because we are running short of time. Thank you very much, Professor Subodhi. It is very lucid and transparent deliberations, and we are looking forward to understand more about the genetic mechanism of salt tolerance. And in fact, it is a very complex character. So we do not expect a very straightforward answer. So there will be many more ideas, and then there will be a synthesis, and then new ideas will come out. So thank you very much for your kind presentations. Thank you very much. May I call upon Endang Aim Sipting C? She is there?
Yes, Septi is there.
Okay, so
good morning, Septi.
Okay.
You can say Septi.
Oh, Septi. Oh,
actually,
not too much.
I hear music though.
Okay.
Yeah, me too, because background noise is there.
Okay, that's right.
There is music in the background, so yeah, could you please?
Yeah, we'll stop it. Now, just to give you some idea about our speaker, she is Associate Professor, Department of Soil and Crop Science, Texas A&M University, Texas, USA. Her current research focus on plant genetics and genomics, QTL mapping, gene editing, etc. And her today's presentation on anaerobic germination tolerance in rice, the underlying genetics and breeding efforts. So this is another very interesting and challenging subject, and I am sure that will create a lot of debate and discussions. With these few words, I will invite Ms. Sethi to deliver her lecture. Thank you very much.
Yeah. Okay, yeah. I'm just gonna prepare my slide first. Okay. So.
Host, please allow her to be co-host so that she can share the slide.
Okay.
Yeah, good.
Can you see my slide? Yeah.
Yeah, we can see now. Yeah.
Okay. Okay, so yeah, thank you for the introduction. So my topic is a little bit different. So this is not about salinity, but about anaerobic germination, especially talk about genetic and also the breeding efforts. So before I came to Texas A&M in 2015, I actually also worked with IRRI, so about 10 years or so. So this also reflected in this presentation. So there is also some connection with IRRI. So before I start my presentation, I like to acknowledge first so the people that contribute to this effort. So the first one is Ranjita Tapa. She's my PhD student, but she graduated in 2019, and now she's a postdoc at Cornell University. Sarmista Gosal, so she's from Biri in Bangladesh. She was my student in IRRI, but then when I moved to TAMU, she then transferred to Dr. Salap Dixit. And then Hidayat Nani, she was my postdoc in IRRI, and then after I moved also, she transferred to work with Tobias Kretschmar and Salap Dixit. And then Dmitry, he helped with the GWAS analysis that done by the IRRI group. And then this is the funding coming from Texas A&M. And then Lee Foundation, this is for Sarmista Gosal PhD in IRRI. And then the MZ, this is for postdoc for Hidya, and also Bill and Melinda Gates Foundation and BMZ for the work that related to IRRI work. So why flooding during germination is important? So this trait is important for both irrigated and rain-fed, and also, of course, include the coastal region that are also prone to flooding. So tolerant during germination is the capability of seeds to germinate and survive under hypoxia or anoxia condition. So this state is important, especially for the rack-seeded system. This is to improve crop establishment, but also important for weed control. And this DSR is increasingly being practiced in Asia due to labor and also water scarcity. And also this DSR is in the US and also in Latin America. This is the mainstay of rice planting. This is how we screen AG in the field in IRRI. Other than that, we also did the screening in the greenhouse. So basically, we sow the seed and then cover with thin soil and then flood the field for about 21 days or so with around 5 to 7 centimeters of water. So Dr. Ismail team and also my own team actually screen for to identify the assess and that are tolerant to flooding during germination. So we identify some of tolerant donors, and we develop a mapping, KTL mapping. And one of the KTL mapping is, the first one is using the Khalanan On. This is a tolerant variety from Myanmar. We identify KTL on chromosome 9, and we have clone this KTL as trihalose phosphatase 7. So here is OSTPP7. It's this enzyme convert T6P become trihalose. So when the seed embryo lacking the T6P or reduced concentration of T6P, so this is sensed by SNRK1, and this is enhance the expression of this transcription factor MIPS, the one that attached to alpha amylase. So this is enhanced starch mobilization that develop more energy here for the germinating seed to elongate the coleoptile and finally reach the water surface. So then they can survive. And this is not happening in the susceptible variety because they don't have the OSTPP gene. And this is just show you the work of Dr. Lal from India. So he used this AG1 nail. So AG1 nail is IS64, the one that have AG1 KTL. So this is these two are the sister line from AG1, and this is IS64, and this is IS64 sub 1, and this is the flooding. So until 21 days, and this is the germination rate. As you can see that throughout the experiment, the sister line having the AG1 have superior performance compared to IS64 or IS64 sub 1. And then the second mapping population, we identified KTL on chromosome 7, and this is coming from Majan Red, which is from China. But we also identified KTL on chromosome 7 in other population using Khalanan On or Nanhi from India, and also Karsu from Pakistan, and TKM9 from India. And also pretty recently, so this is the work of Sarmista Gosal, the PhD student, the one that worked with Salab Dixit. So two mapping population using Kalarata was developed. This is in the background of two elite varieties from the Philippines, which is RC222 and RC238. And the genotyping was done using this CASP assay. And we identified 3 KTLs on chromosome 3, 6, and 7. And these 3 KTL detected in both populations. So this is RC222 mapping population, and this is 238. And this is the 3 KTL that identified in both populations on chromosome 3, 6, and 7. And the one on chromosome 7 was the largest KTL. So this is just the summary of the overlapping KTL that we identified from this 5 mapping population. So, and this is the susceptible parent. I have 42 and I have 64. And we identify the variants in this region, in this detected region. So this is 6 to 13 MB. So it's pretty big because we try to be comprehensive to cover all of the region. And then we try to use the information from the 3000 Rice Genome Project, so the 3K Rice Genome Project. So based on the variant that we identified, we try to identify the genotype that closer to Masan red. So in this case, we identified about 300, more than 300, so 351 accessions. So here there are, so the top here, the genotype is closer to Masan red, and here is less similar to Masan red. And this is just to show, so the 351, this is mostly indica, and some of them are aus. So then we try to measure or try to count the survival under the anaerobic germination in 14 days and 21 days. Okay. Oops. And then, so we identify KTL on chromosome, KTL on chromosome 7, which is pretty significant. And this is much smaller compared to all of the KTL mapping that we identified. So using this GWAS population, we can, this is kind of an enriched population because the accession is mostly similar to Masan red. So we can identify the KTL on chromosome 7 with pretty significant lot. And this position is similar to the KTL that called RC KTL, the one that control the red color. So we kind of wondering if is this the gene underlying this KTL for anaerobic germination linked to the KTL, or actually is pleiotropic with the RC KTL. So currently we still working on this, try to identify the gene underlying this KTL. So in this case, I work with Salab Dixit. So this is, we did some transcriptomic, and then we detect some of this gene. And this is the peak region within chromosome 7. So there are some SNPs, there are some indel, and so on. So I will, I'm not going to elaborate more on the gene identification because we're still working on it. So I'm just going to highlight the breeding that we have done. So when I was in IRRI, I started this MABC with the sub1 and non-sub1. As you know, sub1 is the one that underlying the tolerant for vegetative stage. So we pyramided with, so AG1, AG2 pyramided on the background of RC82, which is an edit line from the Philippines. And then we also pyramided with Cerang sub1. This is, Cerang is an edit line from Indonesia, but I guess Cerang sub1 also released in Bangladesh. And this one is the, so these are some of the performance of the line pyramided AG1, AG2, and sub1. And so this is the IRRI 154 is the RC222, IRRI 156 is RC238. These two are the elite rice from release variety from the Philippines. As you can see, they are susceptible to both submergence during vegetative stage and also anaerobic germination. As you can see, the score here. But we identified some lines Chirang sub-1, Ag1, Ag2 that both are tolerant to submersion and also anaerobic germination, and some of them has better yield compared to the elite varieties from the Philippines. Okay, so other than that, also one of my students here in Texas A&M, Ranjita Tapa, also performed some GWAS using the rice variety from USDA. So mostly coming from Central and South Asia, but also some varieties from Texas and also some US varieties. And we did the seed multiplication in Bowman Research Center. And then we genotype using the 7K SNP array that's developed by Susan McCouch at Cornell University. And we identify, and also at the same time, we also did some transcriptomic study using one of the tolerant lines and one of the susceptible lines. And we use 4D control versus the 4D submerged in the water. And we use Illumina to sequence. And this, we detect some QTL, so, and the one, this is in chromosome 2, for example. So we have some candidates, the one that we thought is kind of potential underlying the QTL on chromosome 2. So the future activities, if this AG will be used for coastal regions, I guess we need to pyramid this QTL with salinity tolerance, other than also with submergence. And for the resident, also we need to explore other traits that are important and can support the robust seeding performance in the coastal regions. So I guess with that, that is the end of my presentation. And this is just my group here in Texas A&M. And also with Dr. Michael Thompson group combined, yeah. Okay, with that, if there are questions, I will be happy to answer, yeah.
Thank you very much. It is a very exhaustive work. And you have taken as many as 268 sessions, and they have more than 80% germination ability. And on that, you have given a lot of experiment, which is modern technology, very cutting edge biotechnological approaches. And I can remember this, you have said about Q12 mapping, GWAS for aging, and then, of course, molecular level, SNP study, and all these things. This is quite a huge domain you have covered. Now, I have got one particular question that you have attempted gene pyramiding. How many donors you have taken? One. And then you have suggested GWAS for aging, tolerance, red color seed. What is the relationship with the color of seed?
I couldn't really hear the second question actually.
Red color seed.
Professor Das,
it?
can you stop the
Yeah.
music? Otherwise, we can't hear you properly. Music is in the background, I think.
That is something, isn't it? Because it is election mode. People are doing that thing. You know, that's the difficulties here. I cannot control it. I cannot control. That's the problem. But anyway, I will try to repeat it. I was wondering about this, that how many donors you have taken for pyramiding, gene pyramiding. And then you have suggested some dose for AG tolerance, red color seed. Why? Is there any relation between the color of seed and tolerance?
Okay, yeah.
I'll
Yeah,
just.
so I try to answer the question. So for the gene pyramid, I guess, I mean, it's not limited if you have, more than one KTL, maybe 3 or 4, you can pyramid it as far as, there is an easy way to do it. But especially if they are in the same background, I guess the same genetic background is much easier. For example, if they are like in, for example, IS64 background, so 4 KTL or something, they can be pyramided. But yeah, in this case, before I moved to IRI, I moved to Texas A&M, we have, so my team has pyramided two KTLs, in this case, AG1 and AG2, but then also pyramided with sub1. So kind of 3 KTLs in this case, yeah. But of course, I mean, it can be, can be pyramided with more KTLs later on, especially if they're in the same background, because then we just monitor the KTL, and we don't really care with the background because they are the same background. Yeah.
Ma'am, I hope till now we are having around 12 or 13 papers only on QTL mapping on like aerobic germination. As much I know, there is not much a study that we are having, and only 3 or 4 QTLs are only like major QTL. So I hope with that only we can go for like improving this trait.
Yeah, yeah.
Just to add on to our PK Das sir, only I have told this one.
Yeah, especially KTL, like kind of stable, detected in many environment, and also many donor, like the one in chromosome 7, that's kind of major KTL, and plus also more stable. So we can work with that kind of KTL. But KTL that, not really stable, only detected in one environment or something, we don't really use that. Yeah. Yeah. And then also there is a question about red color. So is that related to tolerance? If I'm not mistaken, is that the question? Yeah. So in this case, I don't really know yet whether red color can help with the tolerance, but there is a possibility because red color also kind of antioxidant. So antioxidant also help in stress situation. So I don't know if there is correlation about this too, but there is possibility. But I don't know if this KTL the same as RC, the gene underlying the KTL for AG is the same as the RC gene. So I don't know, but maybe even if not, the red color also kind of help just because of the antioxidant that the rice has, because then it can help under the stress also. Yeah.
So one suggestion, the RC gene, actually it is located in the major QTL region for seed dormancy. There are papers, several papers actually. The RC is responsible for seed dormancy. When it is red, it doesn't germinate. So maybe it is having something to do with that. Because in my study also I found that RC gene in that region particularly, there is a major QTL for seed dormancy.
Yeah. So yeah, in this case, we don't really know yet because we haven't really identified the gene for AG, whether there's correlation with it just detected in the kind of the similar region, but we don't know exactly yet because we need to confirm this. Yeah. Thanks, Dr. Subhuti, for the
And nice to see you, Sati.
Yeah.
Okay.
Yes.
Okay,
Nice
Dr. Singh, anything?
No,
Okay.
thank you very much. I think it's okay. I know SEPTI work and SEPTI have since long.
Okay, thank you very much. So it is very interesting topic, and thank you once again. And we'll call for the next speaker, Zeba Siraj and others. It is from Plant Biotechnology Lab, Department of Biochemistry and Molecular Biology, Dhaka University, Bangladesh. She is teaching and researching for more than 30 years. Received Dean's Award for producing salt-tolerant transgenic heritage rice lines in 2013. And Ananya Award 2016. So, and she's actively engaged for quite a bit of time. And her today's deliberations on wild halophytic rice, P. correctata, as a source for rice cultivation in saline soil. And that is, she is going to present. And I invite her to present her talk. Thank you very much.
Thank you very much, Professor Das. Thank you, and thank you for inviting me. I'm thankful to the organizers. So I'll share my screen. Can you see?
Yes, yes.
Okay. So I'll start again by thanking the organizers for inviting me. So thank you for the introduction. So I'm going to speak today on use of, actually many people called it Oryza coarctata as well. Actually more people call it Oryza coarctata. So I changed from P. coarctata to Oryza coarctata. Anyway, so how can we use this as a resource? That's what I'm going to talk about. So in Bangladesh, currently 1 million hectares of coastal soils are affected by salinity. And this is particularly in the dry season. And this area is about 1/9 of the total cultivable area in Bangladesh. So it's quite a lot. So this is a picture of the spatial variation of the salinity. And again, this is usually more in the dry season, because in the wet season, the salt somewhat washes off. So as you can see, the salinity slowly, gradually increases from Satkhira to Khulna to Joshore. And actually this salinity is increasing, going upward. The level is increasing gradually. For a closer look around those areas, so this is like Satkhira and this is Khulna area. So currently the scenario is that rice does not grow below this blue line in the dry season. And the predicted increase in salinity, the worst case scenario is that by 2050, we won't be able to grow rice below the red line. So that is quite ominous. And this will actually translate into a median increase of 26% in salinity by 2050. And rice yield will decline by 15.6%, because in the 9 coastal upazilas, we will not be able to grow rice. And at the same time, the population of Bangladesh is predicted to increase from 160 to 214 million by 2050. Okay, so obviously, we like our rice. So how can we make rice more salt tolerant? So we have some, I'm going to talk about Oryza sativa, and in local language, we call it Uri Dhan. Some people call it Dhani Ghash. Like ghash is the language for grass. So, anyway, so this is endemic to the whole coastal area of Bangladesh. It's also grows in India as well as maybe in Malaysia as well, and maybe further. So with respect to Bangladesh, Oryza sativa, Oryza coarctata grows right from the east tip of Cox's Bazar up to Shatkhira along the coastal line. And this picture, it actually shows, this was taken in Shatkhira, so you can see the Oryza coarctata growing there. And this is in our net house at Dhaka University. This is what it looks like when it flowers. So it flowers sporadically, starting in the monsoon season, say June, and then maybe, once every 2 months it's going to flower. So per panicle, you have only like 10 grains, 10 to 15 grains maximum. Whereas if you compare it with rice, per panicle, you'll have at least 100 grains. So that's the difference. This is how they look like when they mature. So Oryza coarctata actually completes its life cycle in seawater. It can complete its life cycle in seawater, which is, seawater is 400 millimolar of salt. So, it's highly desirable as a resource for, introduction into rice. So it has salt glands. Oryza coarctata has salt glands both in the upper surface of its leaves and the lower surface. And we did a hydroponic experiment where we grew Porteraceae coarctata in hydroponics. And we noticed that the higher the salt that we gave in the hydroponics, the more efficient it desalinated. The rate of desalination was more efficient for Porteraceae coarctata. So, like you can see this curve, which is downward. So we thought that we asked the question, can we use this property of P. coarctata to desalinate and help it grow rice? So that's the question we asked, to desalinate water or desalinate soil. So this work is under progress. So I'm not going to talk about this. In my current presentation, I'm going to talk about the microbial populations which are associated with Oryza coarctata, and also that we cloned some genes from Oryza coarctata and transformed rice with it. So my presentation is going to cover that. So initially, when we started this work with Oryza coarctata, we actually collected the soil in Shatkhira from the fields where the Oryza coarctata is growing. So from that soil, we isolated rhizobacteria and we tested, like single populations of bacteria for their ability to help rice grow. So this work has been published. But then later on, we decided to use a multitude of bacteria, rhizobacteria, which were from the soil, to see whether it better affected, the ability of sensitive rice to grow. So these are some of the pictures for that work. So we found out that this combination J, which are all halo-tolerant rhizobacteria isolated from the soil in Shatkhira where the Porteraceae was growing. And you can see the effect of this combination. So this was rice actually growing in 80 millimolar salt, and you can actually see that the roots are growing pretty well. And from those same plants, we could see under reproductive stress at 80 millimolar salt, the total chlorophyll was significantly improved in rice plants which had been treated with this J combination of rhizobacteria. And again, the yield per plant was also significantly higher for this J combination. So this work is actually still under process. We are, analyzing some more results, and we might publish this work soon. So then we moved on to isolating endophytic bacteria from the Porteraceae coarctata, the Oryza coarctata roots. So, and we isolated only the bacteria, the endophytes, which were very salt tolerant. So we isolated bacteria which grew above 1.2 molar of salt. So I'm going to talk about this particular strain, Staphylococcus petresi, which we isolated from Porteraceae roots. And we also found out that it had certain growth promotion activities, like it had zinc solubilization, and then it had siderophore production. So, and of course, indole acetic acid production as well. So we went on to do some seedling experiments, and you can see that this particular bacteria, endophytic bacteria, it enhances the seedlings under control conditions, or seedling growth under control conditions as well. But when we applied stress, you can actually see that the difference looks significant. And when we analyze the results further, we could see that the shoot weight of these seedlings under stress, they were significantly better than the control, which were, not given any bacteria. And again, when we checked the chlorophyll content of these seedlings was also significantly higher compared to non-bacterized, stress seedlings. So we wanted to take this work further. So we wanted to see the effect of these endophytes on the roots of the rice seedlings. So we actually built some rhizotrons where, we had these glass chambers. So we could observe the effect of these bacteria on the roots. So here, again, when we see there's a positive effect of the bacteria on under non-stressed conditions, and these are the plants in the stress conditions. Here we gave 50 milli. It's not much, but we wanted to test the effect. So what we actually observed is that that the lateral root number was significantly increased in the bacteriized BR2 sensitive rice seedlings. And you can actually see this combination 5. I'll explain what this is. So this is without the bacteria, and this is under stress, 50 millimolar stress, and you can actually see that the lateral roots are higher here, and this was significantly higher compared to non-bacterized seedlings. Okay, so then these results are from a different bacteria, again isolated from Oryza coarctata, which is Micrococcus species. And here we again observed that the shoot length was higher for combination 5, which had this Micrococcus, significantly higher shoot length, and then significantly lower malonaldehyde content in this combination 5. And then again, the chlorophyll was significantly higher, and the survival score for combination 5 was much better for seedlings which had been treated with this Micrococcus endophyte from Oryza coarctata. So we went on to check some genes which reportedly affected lateral root production. So this is the graph which shows what happens. So here what we observed is that if you look at the 72-hour relative quantitative gene expression data for OSIAA13 in root, you can see that in combination 5, you have a very significant enhancement compared to control, that is, non-bacterized. So we are continuing this work, and we want to see some other genes as well, which may be affected. So this enzyme is involved in auxin signaling. The OSIAA13 is involved in auxin signaling, and it controls the expression of genes that are required for lateral root initiation in rice. And we actually also observed that the endophytic bacteria from O. coarctata also significantly increased the number of lateral roots, as I showed, as well as the width of the roots of sensitive BR28 rice under 50 millimolar stress. So we are going to give higher stress, and also we are going to test these results in a reproductive stage. Interestingly, this Micrococcus bacteria, it also caused increase in SOS1. As you know, it's the plasma membrane sodium hydrogen antiporter. And here the fold change was significant for increase for the combination 5 under salt stress, whereas the no bacteria one, there was a relative, no relatively higher gene expression at all for SOS1. And this was at 24 hours. So what's interesting is that, so we isolated this endophyte from Oryza coarctata growing in our net house for more than 6 months after collection. But we isolated some endophytes from, freshly collected samples of Porteracia coarctata. And what we found is that when we did that, the diversity of endophytes which are associated with Porteracia coarctata is much higher. And so these are all actually halophytic, which are growing above at least 1 molar of salt. And just to show you that these bacteria have endophytes from the root of Porteracia coarctata. They have like nitrogen fixation properties, and then they have phosphate solubilization, zinc solubilization, heavy metal tolerance, as well as siderophore activity. And one actually also produced a fluorescent compound, which we found very interesting. So we are going to test some of these, maybe mixtures for improvement in rice. But this slide was just to show you that if Oryza coarctata is growing in its natural environment, more endophytes or more diverse set of endophytes are actually associated with it. Okay, so I'm not going to switch gears again, and I'm going to talk about some of the genes that we isolated from Oryza coarctata and put them into rice. So these genes, when we cloned them, the sequence was already known. So we chose metallothionein, then the abscisic acid stress ripening protein ASR, and the subunit C of the vacuolar ATPase gene from Oryza coarctata. So we use gateway cloning technology, and so we use implanter transformation. So I'll talk about that a little bit more. So metallothionein, we chose that because it's an ROS scavenger. It binds to heavy metals, and it can actually maintain metal homeostasis, and it's highly upregulated under salinity stress. So that was the reason for choosing it. And the Oryza coarctata metallothionein, it has 83% identity to the Oryza sativa metallothionein 3A, and 75% identity to the Oryza sativa metallothionein 3B of rice. And then the ASR protein, this actually functions as a chaperone. It also has a nuclear form, and it's very drought and salt responsive. So it's upregulated in osmotic stress as well as by ABA. So we chose that one. And here, the Oryza coarctata one has only 50% identity to the Oryza sativa ASR1. And I think the Oryza sativa ASR1 is probably, it's not found in all. I think it's, in Pokkali, it has been characterized for Pokkali rice. So only 50% identity, and but they both contain this WABA, WDS domain, but in different locations. So, and we also cloned this vacuolar antiporter gene. So this vacuolar antiporter, of course, it helps maintain negative membrane potential to counteract sodium depolarization, particularly at the vacuole. And this is also significantly upregulated by salt stress. So it's ATP coupled, it pumps protons from the cytoplasm to the vacuole. So as I was saying, that we do implanter transformation, and this implanter transformation actually helps us. We can transform any variety that we want. We don't have to bother about whether it's tissue culture responsive or not. So briefly, the procedure is we soak the seeds for 2 days, and then we inoculate. This is the embryo, just above the embryo or the tip of the embryo. with the agrobacterium containing the construct of interest, and we vacuum infiltrate. We let the seeds germinate in dark for 9 days, and then we transfer it to hydroponic solution, and then we transfer it to soil. So we don't actually wait for, we just move, we collect the seeds at we let the T0 plant flower, and then we check the T1 seeds for transgenicity in this in-planter transformation. So it's transferred to the net house or greenhouse, immediately at this stage, it's transferred to soil. Okay, so this in-planter procedure has been published, and we find this procedure is very good to, transform any variety that we desire. So this is Biridhan 75, one of the newer commercial varieties that Bangladesh Rice Research Institute has released. And we transform it with the PVA gene, the vacuolar PVA gene from Oryza coarctata. So these are T0 plants with the T1 seeds. And again, we transformed Biridhan 81, another desirable commercial variety from Biri, with the ASR gene of Oryza coarctata. So these are leaves from the T1 plants, and as you can see that the ones which were transgenic, this is for the PVA1 gene in 150 millimolar. So we did like this is not loaf, it's leaf disc senescence assay, and you can see that these are green after about 5 days, whereas this is the wild type BR75, and this is wild type in 0 molar sodium chloride. And then this is for the ASR gene. Again, we can see that these still remain green after 5 days in 150 millimolar sodium chloride. This is the wild type without salt, and this is the wild type in salt. So these we are up, like T2, or we are going up to T2, T3 generation before doing the whole, bunch of screening studies at seedling as well as reproductive stage for tolerance. And the metallothionein work it's still progressing. So we are very much interested in the calcium ATPase gene of Oryza coarctata, because we found that it continues to, its expression continues to increase right from lower salt to higher salt concentration, and this is a gradual increase, and it's both in leaf and root. Whereas we found that for the NHX1, the vacuolar antipoter gene, only increased there was a gradual increase only in leaf, and for SOS1 gradual increase only in roots. But this one, it seems to increase both in roots and as well as shoots, and it might be something to do with signaling of calcium stores when when sodium comes there. So we found it very interesting. So these are our results that we got from Oryza coarctata, quantitative real PCR studies, quantitative, and we found that there's a gradual increase both in roots as well as in shoots. So we are trying to clone this. It hasn't been successful yet, but we are trying to close this calcium ATPase gene from Potaresia coarctata. And from papers on calcium ATPase, as I said, it appears to signal release of calcium stores immediately within like a few seconds of salt stress. So we are going to try to clone that. So interestingly, the Potaresia coarctata calcium ATPase has a lot of deletions, significant amino acid deletions, and one of my students just did some homology studies. So this is the Oryza sativa calcium ATPase, the Oryza coarctata calcium ATPase, and this is the Oryza sativa one. So the Oryza sativa one seems to have some additional, groups there in that particular region. And when she superimposed both of them, you can see that this is the additional domain that the Oryza sativa calcium ATPase has, but we don't really know what the role of this particular, why the Potaresia coarctata should be more efficient because when we, obviously for the real-time PCR, we did the Oryza sativa as well. And although it increases, but not beyond 100 millimolar for the Oryza sativa one. So what role this extra has, or the absence of this has for the Portyressa for the Oryza coarctata calcium ATPase gene, we don't know yet. So that's about what I wanted to talk about today. And this is my team and the students who are working on this endophyte and rhizo. So this is Pratap Kumar. He did the rhizobacterial work. And then Dola Kormokar, she has been working on the endophytes. She's still working and writing some of the papers. And Onik is he just finished his master's, and he's been doing the Rhizotron work, and he's trying to look at a lot of software which will analyze the root structure for us. So he's experimenting into that. Dr. Rakibul Islam, he's a microbial expert who helps us. So, and Habiba Mita, her PhD is actually on Oryza coarctata, and she's currently in IRRI. So most of the cloning work is hers, and so that's why the cloning work is a bit slow when she comes back. So she's doing part of the work in, part of the work in sort of characterizing more the Oryza coarctata from Bangladesh and comparing it with other accessions, and also doing some other work on Portyressa coarctata. So thank you very much for your patient hearing, and thanks again for inviting me.
Professor Das, you are mute.
Right. Because background noise is too much. But anyway, very exhaustive study it is, and very interesting study. Looking at these results, which tempt me to suggest one more thing, that these coarctata genes, can it be useful for pollution control? What your opinion about it? Because heavy metals and other things we are talking.
Yes, yes. So one of the problems with Portyressa coarctata is that it takes,
It is mute. We cannot hear it.
Oryza coarctata, it takes time to establish itself. So it might take, 2 to 3 months to establish itself in soil. So that's the only problem. But once it grows, it actually becomes very weedy very quickly. So you have to control it. And, because it spreads from its rhizomes, it spreads, it spreads a lot. So I don't know, we're trying to use it for desalination. So those experiments, we are still conducting them. So there's a high variability, sometimes it just doesn't like to grow Portyressa coarctata. But once it does, it kind of takes over. So, so you have to both control its, how it grows. So that's the main problem with controlling Oryza coarctata.
Dr. Singh,
thank you very much. No, it's okay from my side. Dr. Zeba, just to know that in case you are also looking for like a coarctata for like a, for the like a heavy something like a, actually when we are talking about the coarctata, we are talking about the tolerance level increase. Yeah, I cannot. There is a noise. Okay.
Yeah, we can mute it.
Yeah. So when we are talking about the coarctata, because it's halophytic in nature, and we are just marrying halophytic with the glycophyte. So,
yes.
So does it also not having like a possibility that in case if you breed a rice which is having the multiple genes from the coarctata, and it becomes something like a salt loving something like that, you know. Could you get my point? Means like if it needs salt to grow better or something like that. Do you think?
No, it doesn't actually. It grows perfectly happily without salt.
Okay.
Yeah. So you can call it a facultative, you know, it's not an obligate. It's not an obligate halophyte. Yeah.
Okay. Thank you.
Yeah.
I have one question, Dr. Jeva. How much
yes.
the efficiency of implanter transformation?
Oh, very high efficiency. Almost like 30%.
Well, that's
very high. I'm very happy with this technique.
Oh, okay. That's and the bacular ATPase, how it have you compared it with Spartina alterniflora?
No.
Because that's a halophyte. Actually, it's grown in Louisiana in coastal areas.
Okay. No, I haven't. Actually, sorry, like most of our projects, we don't really have a lot of PhD fellowships and stuff like that. So most of this work is started by MS students, and they don't have a lot of time. So these genes were available, and the whole sequence. So the coding sequence. So we just proceeded, and ultimately this girl Habiba, she started her PhD on that. So I'm very much interested in the metallothionein transgenic, see how it works out. So, but that one is a bit slower, and we're looking forward to the calcium ATPase. So hopefully we can clone it.
So it is not, because I know Dr. Zena did some work with crossing with rice.
Yes.
But it's very hard to get.
So interesting. I didn't report on that because I haven't published yet. So the thing is, what we did is we took one of our local varieties, Latishal, and we made it into a tetraploid. And then we actually crossed with Porteraceae, right? So we have hybrids which look like rice, right? But they do not resemble either of the parents, more like rice. So we are continuing to work on that. And as I said, Habiba, she's trying to characterize these putative hybrids. I don't know if they are, but somehow it seems to me like, like the bulbosum technique where, you cross Hordeum vulgare with the other one, and then there's a chromosome loss. So I think that's what's happening. But this cross was between, a 4N Latishal and PC coarctata, and always the PC can be the mother, but we use the 4X Latishal as the mother, and we produce these which look like rice, but they're not. It's not selfing because the hybrids grow very slowly and everything. But anyway, we are trying to characterize what these are. So hopefully she's going to do some chromosome studies and see what's happening. So, are there any fragments? But we don't, in these, whatever they are, we don't really get the salt tolerance. So we have to find devise some other way of getting the salt tolerance into those. Maybe, grow the plants under salt, the plants that we are using for crossing or something like that, so that, but we haven't found, we do get slightly increased salt tolerance, but it's not significantly higher. It's not higher than our transgenic work or any of the others. So the transgenics are actually better rather than these putative hybrids. So I don't talk about it because it's difficult to prove. And so we are trying to do that now. So anyway.
Okay, thank you very much for your nice presentations. And
thank you.
Okay, and once again, thanks, because we're running short of time, and I'm sure that it has generated quite a bit of inquisitiveness. People are trying to know much more about. But anyway, the next time probably we'll be able to interact much more. Thank you very much. Now we have got at the end we have got one lecture and it is going to be delivered by Mitchell J. Thompson. Is he available right now?
Yes, he is there, Professor Das. After that, there will be one more lecture of Dr. Veena.
Okay. But we'll just proceed with Professor Thompson, right?
Yes.
Yes, correct.
Okay. Now, welcome, very hearty welcome, Professor Thompson. He is the Professor, Department of Soil and Crop Science, Texas A&M University, Texas, USA. And he is a leading agri-life research genome editing lab and working to optimize CRISPR-based editing approaches and trait development in crops. So it is a very interesting subject he is handling, and today he is going to deliver his lecture on biotechnological approaches to develop salt-tolerant rice for the coastal regions. And it is a biotechnological approaches, okay? And so it is in fact, a review on current status of molecular breeding tools and explore future directions. So with this, I would invite again Dr. Professor Thompson to deliver his talk. Professor Thompson, please.
Okay, thank you very much, Professor Das. And let me just share my slides. So hopefully everyone can see them now.
Yes.
So yes, it's really my pleasure to talk about, give an overview of biotechnology, as it's used for salt tolerance. And, this work started quite a long time back when I was at Erie for about 10 years. And then for the last 5.5 years, I've been at Texas A&M University. So just to kind of, as an introduction, I mean, most of my research touches on 3 different areas. And one of them is genetic resources, the other is plant breeding, and then also plant genomics. So I've been working on how to combine, these 3 areas and what's the most efficient techniques to do that, and also as it's applied towards salt tolerance. And so really the starting point is we have this, what we call a genetic toolkit for modern plant breeding. So the starting point really is genetic diversity. So we have crop genetic resources, and we've already heard several talks about looking at wild relatives or exotic germplasm or land races. And then on top of that, we have really a lot of basic research that's going on. And so many, research projects into gene discovery, now allele mining. Many of these started off with gene mapping, QTL mapping, and then genome-wide association studies. And then, next-gen sequencing and other techniques have really accelerated that process. And so I see kind of 3 different applications or approaches to use all of this valuable data and genetic resources. And so the first really is marker-assisted selection. And so there's, this has been used for a long time, and more recently with high-throughput SNP genotyping to select for, say, large effect genes for your trait of interest. And of course, that's a non-GMO product in the end. And then next is genetic engineering. So we've heard from Dr. Zeba Siraj about, you can use transgenics to really improve your traits. And then I'll also talk a bit about genome editing as well. So newer technology using, CRISPR, Cas9 systems to make precise changes in genes. And it turns out that you can have, gene editing products. Some of them are GMO, but some of them can be non-GMO. And so it kind of fits right in between, more of a DNA marker selection technique versus, pure transgenics and genetic engineering. And so I'll try to touch on those today. But I think, many of you are familiar with salinity tolerance, and we've already heard a bit about it today. And so just real quick, I mean, we know that there's many different molecular mechanisms, controlling salinity tolerance, and this could be sodium exclusion or detoxification, compartmentation, osmotic protection. And the idea that I want to kind of get across is that once we know, the genetic loci and the genes controlling these particular trait components, we can then work to use these for breeding and for crop improvement. And so I'll talk about different ways that we're trying to understand, at least in rice, what are all the genetic components controlling these. And again, the starting point is genetic diversity. And so I was back at IRRI. I was there from 2005 to 2015, working initially on salinity tolerance under Dr. Abdelbagi Ismail, of course, with collaboration with R.K. Singh and Glenn Gregorio and others. And we had a student, Aklas Rahman, who was looking at genetic diversity. And the idea here is that we have a number of known salt tolerant donors. And then he wanted to compare, he had some kind of interesting germplasm from Bangladesh that was salt tolerant, and he wanted to know what's the relationship between this new germplasm with some of the conventional donors for salt tolerance, such as Pokali or Nono Bokra and others. And so using, in this case, 384 SNP markers, we can then distinguish subgroups within indica, and then it's very obvious that AUS is actually a very separate subgroup, and then aromatics and others, to determine where, for instance, he identified a salt tolerant variety called Kapsul that's known in Bangladesh, but at that time we didn't know what's the relationship to Pokali and others. And so that's one way to determine, to identify potentially novel genetic donors. And some of the background is, of course, this famous population, from IRRI that, started way back with Dr. Senadiran, Glenn Gregorio, and then again, Dr. Ismail and R.K. Singh and others, looking at IR29 by Pokali, because IR29 is very, very sensitive. Pokali, of course, is tolerant for some of the mechanisms of salt tolerance. And of course, there were many QTLs, but one, the biggest one was saltol and chromosome 1. And so that was the focus of a lot of effort. But I'll still kind of highlight that these other QTLs are also quite important. So when we see something like FL378, FL478, the effect, that tolerance is not just from a single QTL, it's actually from many QTLs, that combined effect. But again, there's a lot of effort, going into that saltol QTL. So here's one of the kind of early studies that we did trying to use this kind of substitution mapping approach. Again, some of these different RALs had different introgressions across the saltol region. And it was kind of interesting also that some of them even had different alleles. And so it looks like there's, it wasn't a simple matter of just, one tolerant allele, one sensitive, that you actually potentially have several different tolerant alleles at that locus. And of course, I know people have, taken this and tried to integress it, but some highlights here is that you do have to be careful which donor you're using, because they do potentially have different alleles. And then you just probably will need more than one QTL, because it's not just, saltol that's going to give you a major effect. You'll need multiple QTLs. And that brings us to, some of our early work back then was on comparative QTL mapping, looking at, the boxes are QTLs, and then, some of these known genes that are known to be involved in salinity tolerance, such as, this SKC1 or these different HKT genes or NHX genes. So many of these, of course, are mapped in rice, and then you can kind of correlate, the QTL locations with these major genes. But it's not just, from Pokali, there's many other tolerant donors I mentioned. So this Aklas Rahman published a bit more recently his capsule work, where he identified some major QTLs from that Bangladesh salt tolerant variety capsule. And so these then also can be used, for marker assisted selection. And then more recently, so I had a collaboration, a few years back when R.K. Singh was still at IRRI, with sending one of our graduate students, Sujuti Mandal, over to IRRI to do work with reproductive stage salinity tolerance. And there was a question earlier about that, and it is much, much more difficult than seedling stage to map QTLs for reproductive stage. But she did a lot of work at IRRI, and then coming back, doing genotyping back at Texas A&M. And it was a successful collaboration. So she used salt tolerant parent CSR28 and Hasawi, crossed with Biridon 28 from Bangladesh. And again, so all the population work was done with Dr. R.K. Singh and the phenotyping back then at IRRI, and then the genotyping with 2 different systems. So CASP genotyping and then whole genome skim sequencing. Here at Texas A&M, it's similar to a GBS type approach. So you get a much higher resolution, but you also need much higher quality DNA and it's more expensive. So there's some drawbacks to that as well. But she did find QTLs for a number of filled spikelets under salinity stress at the reproductive stage. And so again, these are, provide information that could be further, investigated to see, are they, worth pursuing in a breeding context, or potentially trying to identify, what are the candidate genes in these regions. But then since then, she's since graduated. So then I want to kind of switch over to talk about gene editing. So there's already been, some discussion of, how can we use gene editing for breeding? And certainly I think it is very promising. So I wanted to kind of quickly introduce, what is the technology? And so it started off back in really 2012 when Jennifer Dunia and Dr. Charpentier, this is a review from 2014, but in 2012, they published their work showing CRISPR-Cas9 can be used very easily for targeted gene editing. And of course, that won them the Nobel Prize in chemistry last year. And so this is, again, very important technology. And just to real quick, for those who are not as familiar with it, it actually, the CRISPR system is a bacterial immune system from viruses. And so it's the way bacteria protect themselves from bacteriophages. So these viruses will try to invade. The bacteria will actually capture some of that viral genetic sequence and integrate it into their own genomes as kind of a memory of previous infections, so that the next time that bacteriophage tries to invade, the bacteria will very quickly identify it and then cleave, that DNA or even RNA to destroy the virus. And so that comes from an acronym, clustered regularly interspaced short palindromic repeats. So that's where CRISPR comes from. And Cas9 is CRISPR-associated protein 9. But there's actually many different nucleases now that can be used. And so there's many different CRISPR systems in bacteria. But for our purposes, really, we just need to know the end effect, which is a double-strand break. So the CRISPR-Cas9 will cleave the DNA at a specific location. And then the plant repair mechanisms will use either non-homologous end joining, or NHEJ, or homology-directed recombination, or HR, to repair that double-strand break. And so then you can potentially have a gene knockout because that repair process is quite error-prone if it's NHEJ, or you can have gene insertions. And then more precisely, if there's a template that's used, it can make very precise modifications. And so that's where we get, precise gene editing, either knockouts or modifications or insertions using this technology. And so coming back to, how can we use that technology? And so I've just highlighted a few kind of ways that people are thinking about using CRISPR. One is making exotic germplasm more readily usable. So one issue we've always had, back when I was at IRRI, is how do we use a genetic donor such as Pokali or these other, say it's highly salt tolerant, but it has a lot of negative characteristics. So there's a lot of effort to improve, the genetic donors themselves so that they can be more easily crossed with breeding materials without, bringing all this kind of negative linkage drag. And so essentially, one way, you might do that actually is, instead of trying to take Pokali and extract, the salt tolerant genes, one way you might think about is, in a way, kind of taking Pokali and removing all the negative characteristics. And so there's, during domestication process, and when you improve germplasm, you're essentially removing negative characteristics. So with gene editing, you can more rapidly improve genetic donors using this kind of reverse technique. But other ways is you can identify useful alleles at genetic loci, do direct transfer of these alleles, or you can use that knowledge to design, specific base edits or insertions. And so you can have then these either a single gene or multiple gene edited products. Okay. But just to kind of highlight that example I talked about. So for instance, if you do look at like Pokali, a modern breeder would have a hard time incorporating that directly into their breeding program. So you may have like awns or black holes and red grains, maybe very tall and late flowering. But actually, most of the genes for these traits are known. And it would be relatively simple to use CRISPR-Cas9 to knock out, say, the gene for awns or, red grain color or, make it earlier flowering and semi-dwarf and things like that. So that's, where that comes from is it's often easier to knock out a gene than it is to, say, knock in or introduce a more desirable allele at a certain locus. So that brings us, to my current research at Texas A&M. So as Dr. Septi mentioned, we have a crop genome editing lab, and we're both kind of leading that effort and essentially developing a high throughput gene editing pipeline. And we're using rice kind of as our model system, but we're also applying it to other crops as well. I'll just highlight a couple, techniques we're using to kind of improve the CRISPR delivery and gene editing systems. One is, protoplast isolation is actually quite useful now. I know people have been working on this, for 30 years, but there's kind of a renewal of interest in protoplast isolation, mainly because you can do transient gene editing to test your guide RNA function, but also you can think about, possible regeneration to enable really more large-scale gene editing, because it is actually the transformation, the regeneration bottleneck that's the biggest really challenge right now. And so I know there's actually a number of groups working on improving the protoplast regeneration process. But again, that's just one possible solution. There's many others as well. And then we're also looking at testing new delivery approaches. So instead of agrobacterium or the gene gun, one approach we looked at is actually electroporation using these plasma jets, which is essentially high voltage with a gas such as helium gas at room temperature to again kind of deliver DNA into plant cells or callus material. There's also nanoparticle based delivery. So there's a lot of new techniques now that the idea is if we can, again, something similar to the implanted transformation that Dr. Zeba Siraj mentioned, if there's a way you can bypass, that lengthy tissue culture and in vitro regeneration process, that would probably be ideal, essentially. Okay. So I'll just highlight, just one project that, again, this is started by Sujuti Mandal, but then she graduated, and so we'll continue it with someone else. But essentially, the idea of gene editing is you can edit not just one gene, but actually multiple genes at the same time. So if you look at something like the HKT family of sodium potassium transporters in rice, there's at least 7 different members of this gene family. And so you might think about, okay, how are we going to characterize the gene function of each of these? And it's quite difficult to get a knockout of all 7 using kind of traditional, mutagenesis approaches. And so with gene editing, it is much easier to do multiplex knockouts. So one idea is that you can knock out all of them or all of them minus 1 to see, what's the function of a particular gene member of this family. And so that's one potential project, again, that's kind of ongoing. And then I just wanted to kind of end with a mention about the regulations. So in the US, USDA APHIS has published this new rule where they do provide exemptions for gene editing. At least in the US, you can get an exemption if it's a simple deletion, if it's just a single base pair substitution, or if you're just introducing a known gene from the same gene pool. So from one of the, from the same species without any modification, if you just simply transfer that gene. These 3 then allow an exemption, so you do not have to go through the deregulation process. So it's much easier to get it, into the field. But I will mention that, of course, farmers are very hesitant to use this technology, especially in rice and wheat in the US, because there are no, transgenic rice and wheat. And so they're worried, I think, more about the export, say, to Europe than they are about, the approval itself. And so I think this is helpful. It's moving in the right direction, but there's still a lot of hesitancy to use, this technology. And just my last slide here, just some ideas on, someone mentioned like haplotype selection and breeding. There's a bunch of data from the 3000 Rice Genomes Project from IRRI and China working on, identifying what are all the different alleles at the major genetic loci. And we know a lot of genes in rice. And so how are we going to really move to the next step? And I think that's identifying functional nucleotide polymorphisms, and then using those kind of haplotypes either for marker-assisted selection or specifically for gene editing to improve, the alleles at each of those loci. So with that, I hope I've shown that, decades of research has actually identified many of these genes in crop plants, and we're hoping to use those for accelerating crop improvement. And one way to do that, I think, is gene editing using CRISPR-Cas9. But really the regulations are more clear, and then in the end, it's really consumer acceptance that's going to be essential moving forward. So with that, thank you, and I'll answer any questions.
Thank you, Professor. Very informative and overview of all this scientific thought going on all over. And actually, you started with the genetic diversity, then went on to mining, allele mining, gene mining, and then, of course, different technologies, modern technologies, QTL mapping, and then gene editing and all these things. You have given a very exhaustive but very precise view. Now, I was just wondering one particular suggestions you have made, that is nanoparticle-based delivery system. So if you produce the protoplast and use that nano-based delivery system, what should be the prospect of having your gene insertion?
Yeah. So that's a good question. And I think, we've seen there's publications out of UC Berkeley where they're using actual like carbon nanotubes and other nanoparticles. And I think, you've mentioned one application, for instance, protoplasts, you can very easily, transform those. But I think even more important would be, going directly into like, plant transformation, like say like a more implanta approach. And so essentially you can attach, the DNA plasmids to the outside of these nanotubes, and then they can essentially enter the plant cell. So it's a way, I think, to avoid, say, agrobacterium technology. So some regulatory systems don't want to see agrobacterium, and so I'd say it's a more kind of neutral approach where you don't need to use agrobacterium for the transformation.
Thank you very much. Dr. Singh?
Yeah, actually, it was a very nice presentation, and some of the places in the LNT we were together. But on the CRISPR-Cas, actually, you already gave the overview of the regulation, because that was in my mind. I knew that one of the variety of the wheat, it was approved in Brazil. It was from the US or somewhere, I don't know, or somewhere. It is the first variety or first crop, which is like for the food. So people are hesitant about that. So CRISPR-Cas, I know that there are like a mushroom color, etc. There are already non-GMO in the US. So if we take it as in the rice, will it be considered as GMO, or will it be having some reservations on that? Because this is a direct consumption of the food, something like that.
Yeah. And I think the technology is non-GMO. If you even if you use agrobacterium, you can segregate out the Cas9, you can get rid of the integration and just keep the mutation. But in people's mind, I think, it'll be very easy for them to consider, gene editing as GMO is the same thing. So I'd say for scientists, for us, of course, non-GMO, but when it gets to the market, people, I think, will just lump them all together into one bucket. But so it's hard, I think, for the consumer acceptance, like you mentioned. So.
May I make a comment on this?
Please,
Mr.
sir.
Chairman. Yeah, I think there has been a separate regulatory policy makers who will take care of the non-GMO, at least the gene modified crops. They are to be considered as a non-GMO, And the restrictions that you have in case of transgenic plants is a marker which is there, the selectable marker. In case of genome edited, you don't need the marker. So that's another thing that one has the advantage of the gene modified or genetically modified genome edited plants. And I believe throughout the world, some new policy are being drafted now in comparison or in contrast to the GMO regulations. Even in India, we know of committees which are trying to do the genome edited plants and genetic engineering engineered plants. There are two different types of things that will come. And definitely one can do it very precisely rather than doing it in a transgenic system where you do not know the site of integration. You have to know the site of integration after you make the transgenics. That's another part that we need to take care of.
Yes, sir. This is true, sir. But you know what happened, that many the international institutions, they have to go for the like a disclosure policy. I know that after cleavage of that, you know, unwanted and like a particle or something, even, you know, knockout mutants, you don't enter, don't put anything on that. But when you are disclosing the things, it is, as Mike mentioned, in the people's mind. Even sometime I remember that many people ask about the marker assisted selection, also GMO, when they say, oh no, there is nothing to do with the GMO and the marker assisted selection. So this is like one step ahead of that, and then people are really very difficult to convince them that this is not GMO.
Right. You're absolutely right, Dr. Singh. It's a question of perception. How do you live at it? And I'm sure this will go away very soon. The advantages of
probably in our lifetime.
Yeah, may not be our lifetime. Maybe in some other lifetime. It will be for the humanity. Yes, sure. Thank you, Dr. Johnson.
Thank you. Yeah.
Dr. Das, you are mute. Professor Das,
right, you are. Okay. Thank you very much, Professor Thompson. It is really very interesting subject you have touched upon, and very debatable, and a lot of discussions going on all over. And it will continue to draw a lot of criticism, at the same time, a lot of praise. So this is all about science, that at the beginning, the people will not accept, but when they will find it, and then they will probably go for it. But it is a long time process. But anyway, being a scientist, we have to carry forward what are the opportunities and advantages we have to utilize them. Thank you very much for your beautiful deliberations. And once again, on behalf of myself and behalf of the other co-chairmen and organizer, we thank you very much. Thank you.
Now,
so,
last student,
it is mute?
No, last presentation from Dr. Veena now.
Oh, last presentation. Okay. Okay. So we'll ask the morning session she has to present, isn't it?
Yeah, correct, sir.
That is actually. Okay. Now, the technical hitch is over for her. She is okay. Now I'll request Veena Devi to come forward to deliver her lectures. It is a rice research station, Kerala Agricultural University, Kerala, and her topic, SSR marker analysis for the identification of the elite rice variety, Lavanya, with the current GTL3 and DNA fingerprinting. So, Veena Devi, please.
Thank you, sir. A very good evening to one and all. And first of all, I thank every one of you to permitting me for this presentation in this late hour. I promise I won't take much of your time. And actually, this is a work. This is only a part of our work that is in the development of our one of the latest variety, that is Lavanya, which is the 10th rice variety of the VTL series. And my topic topic is SSR marker analysis for the identification of this elite rice variety Lavanya, which we developed through mutation breeding with its parent VTL3, which is a tall indica variety, which is otherwise, though it is a tall one, it's prone to lodging, but otherwise it is a very popular variety in Pokali region and the DNA fingerprinting. We go it. We'll do this. As you all know, though the rice cultivation in Kerala is very less compared to other states of India, in here we are cultivating rice in a wide range of agroecological zones, right from the below mean sea level cultivation in Kuttanad to the high ranges of Wayanad. Thus, we are having a very rich genetic diversity among the rice varieties. Apart from this genetic diversity, these varieties possess several adaptable features like resistance to various biotic as well as abiotic stresses. However, all these indica varieties, which are tall nature and are prone to lodging and resulted in heavy crop loss. By keeping this in mind, we started to develop semi-tall rice varieties, which is non-lodging. And several researchers have reported successful uses of induced mutations to improve characters like plant height, maturity period, that is duration, as well as various grain characteristics. And so many DOF mutants of many local varieties have been produced in many countries. So keeping all these things in our mind, we also started mutation breeding and tried to reduce the height of our otherwise tall varieties. There is normally in Pokali varieties, which are very tall compared to even among the indica varieties. I think they are the tallest one, which is grow up to a height of 2 meters. So we try to reduce its height. And in this study, we found that gamma radiation with 40 kiloracks was found effective in inducing the semi-dwarfism of our variety VTL3. Since mutations are random changes in the genetic structure, it is very essential to check whether the salinity tolerance, because in rice, as you all know, in rice resuscitation vitella, we are dealing with the saline tolerant varieties, and we are developing varieties especially to Pokali areas. So these, whether this salinity tolerant gene is unaffected in the, we have to make sure it is unaffected in the developing lines. So with these objectives, we screen the mutant rice varieties, mutant lines of VTL3 with saltol linked markers, and later, after selecting the promising one, we have to fingerprint the variety, covering whole genome, rice genome, for distinguishing that particular variety from its parent VTL3. So this was our study, and the first part we screened the all the mutant lines for the presence of SSM, presence of saline tolerance saltol QTL, and later we fingerprint
Not audible.
She lost the connection.
method we used where we, for the DNA isolation, we use simple CTAP technique. Can you scroll the? So am I audible?
Yes.
Can you scroll the next slide?
1 minute.
For genome DNA isolation, we have extracted the total genomic DNA from the young plants of 3 weeks old plants following the simple CTAP technique, and later PCR amplification was done. And data scoring, the scoring of alleles for each SSR marker was done to find out the total number of alleles as well as the allelic data, and later converted into binary data, and PIC, polymorphic information content value for each SSR marker was calculated as per Anderson et al. 1993. And in the first part, as I told, we were screen all the mutant lines using the for the presence of Saltol QTL. So these 14 SSR markers, specifically for Saltol QTL, were used for the screening of mutant lines. And the following are few molecular profile pictures showing various markers and various lines. And from these pictures, it is clear that the saline tolerant gene was unaffected in all these lines. So for the next, and for the second part of the work, that is the DNA fingerprinting, to identify the particular variety, that is Lavanya, from its parent, we used nearly 48 SSR markers, that is 4 markers each, 4 to 5 markers each from the each chromosome. So 48 SSR markers were used, and we found that 8 among the markers, 8 markers were found to have polymorphic in this particular variety. And as conclusion, we could say that the semitol mutant of popular cultivated Saltol rice variety VTL3 was developed using gamma radiation, that is 40 kiloradians, and later released as VTL10 and named it as Lavanya. And this variety is short in stature and is on screening with key markers linked to a Saltol QTL. It was found that the region was unaffected due to induced mutagenesis, and on genome-wide DNA analysis with a SSR marker based DNA fingerprinting, we could generate a distinguish the particular variety from its parent variety, and 8 SSR markers can be show particularly polymorphism in this particular variety. That's all. Thank you.
Thank you very much. Now you have got this study, what you have suggested, and did you have any validations in the field experiment that your mutant line, which you are saying that genes are not affected? So in the salt tolerance study, what they are performing? Any validation?
Yes, sir, we have even field we proved that it was unaffected, and now it is gaining popularity among the farmers, sir.
Thank you. Thank you. Dr. Singh?
Yeah, thank you, madam. Actually, I wanted to know that up to what salinity it can tolerate, up to, like, for particularly the reproductive stage.
So actually, our Pokali is having seedling stage salinity. But in Kerala condition, as we are cultivating, rice cultivation is undertaking in Pokali regions only during the first crop season, that is in the monsoon season. So in the flowering stage, salinity was not a problem. At present, it is not a problem, but now as the rainfall monsoon is not becoming unpredictable, it is gaining important, that is the reproductive stage tolerance also. And now I am concentrating on those in that line of work, sir.
So do you feel that this is having the similar tolerance to the Pokali, or having the more or less?
This is having similar tolerance to Pokali, sir. But this variety is particularly having very high tillering capacity. So
And how about the quality, grain quality? Is it a
Grain
red
quality
rice?
is very good, sir, and very good. And our farmers are already accepted the variety, sir.
Okay, great. Thank you.
Thank you very much. We are running late, really. It is quite more than 8 o'clock, and we are supposed to finish it by 7:30 or something like that. Anyway, so it is my humble duty to express our thanks to everybody who has participated, and it is a very educated experience. And since morning, we are listening so much of knowledgeable people, and it is, in fact, it is very educative for me at least. And I thank profusely organizers to organize such a beautiful galaxy of learned people who are ready to explain their thoughts, and that way I'm sure the idea of salinity mechanism will be further elaborated. And since it is a very complex trait, it is not possible to solve the problem in one go. It is needed a multiple approaches, right from conventional down to the cutting edge technology. And if there is a synergistic approach, then it will produce a very fruitful and productive result. And as you know, that India, we have got a very peculiar situations. 6.7 million hectares are affected in India. And coastal areas, less than 10 meters above sea level, 600 million people. And within 100 kilometers of the coast, nearly 2.4 billion people. And naturally, we are in a great risk. Under the climate change conditions, we have to face the threat much more. And you know that most part of coastal areas, rice and fish is the main staple food. So since long we are talking about rice in the morning down to the evening, and hopefully we'll be able to come across with some of the very beautiful results and to solve the food security and nutritional security, and which is the present theme of this whole exercise, whole seminar. And I'm very sure that everybody has enjoyed, and they have given a very patient hearing, and they didn't bother to answer any tricky questions. They are always exposed to the new ideas and thought. And I am hopeful that with this approach, in near future, we will be able to come a very meaningful, productive output. And I'm sure that organizers for last another 2 days, there will be great deliberations. And in fact, whatever we have discussed today, it is a crop improvement side, but at the same time, there will be a management side. And when these two together, then there will be a great performance. But as I always think that 3 P's are very important: policy, planning, and performance. So if there is a very strong policy and international and national network is effective, then this kind of problem can be solved, and hopefully can be solved. We are trying to do that, and for the scientific community, our thought is that to advocate and put forward some of the ideas before the policymakers so that they can take proactive role. And unfortunately, now they are thinking, but though it is late, but they are coming forward, and many more deliberations going on, and very strong political will is needed to have a real performance. One of the very interesting difficulties here, I find that production can be encouraged, can be accelerated, but that is a farm level we are talking about, not about the farmers. Their income generation is very important. So that linkage has to be created. That means production and the economy, both to be integrated. Then only there is a possibility that we can address adequately farm and farmers. So it is a high time we must take into action and thought about not only farm, but also farmers as well. With these few words, I thank my co-chairman and other organizers, rapporteurs, and the participants. It is very interesting and thought-provoking sessions. And I would request Dr. Singh to say a few words.
Thank you, sir. Thank you. You already very well summarized the whole session and also give the gist of that with the 3P also. And actually, this is already very late, as you mentioned. I don't want to linger on much on that, but definitely our session was very salty right from the beginning. We had a taste of salt right from the morning. And also, as you mentioned, rice and fish is the culture of the coastal area. Definitely fish must be somewhere in the different session. So we have to mix together somewhere the rice and fish together to make a good food and maybe supper and dinner. And I profusely thank Professor Das and Dr. Professor Majmudar, that they also conducted the session very nicely. And actually, it was really thought-provoking. And also we had a very good issues particularly, and those could be one of the good issues for the younger generation to just take up. That's the main thing, you know. You have to learn that, because you need not to reinvent the wheel, but you have to just take from where we are leaving that wheel to develop. And with this, thank you very much for Nitish and also our reporter, Dr. Manohar. And with this, thank you very much. I don't want to take more time because it's a dinner time for you people.
Just a minute. Actually,
Professor Majumdar is there? Laide Majumdar?
No, sir.
No, he is not there. Yeah.
Okay.
On behalf of ISCAR, Indian Society of Coastal Agricultural Research, I want to thank the chairman of the session, Dr. P.K. Das. He has very nicely conducted the all sessions and all the lectures and these all things. I'm highly indebted to him. And I also want to thank the co-chairman of the session, Dr. R.K. Singh sir, and Dr. A.L. Majumdar sir, and and they also has helped Dr. P.K. Das sir in highly nicely conducting, and we had very great kind of discussion. And I want to thank also all the invited lecturers. They have very nicely contributed, very nicely presented, and we all have really enjoyed. And personally, what I want to say here is that I'm like very new to rice. I'm also a plant breeder, but I'm very new to rice because I have worked in maize, I have worked in chickpea, but rice is very new crop for me, hardly 6 months old. And within this 6 months, how many papers I have read in rice, I'm very nicely and I'm feeling very happy to interact with all those persons, all those peoples who are here. And thanks for that also. And at last, I want to thank all the participants, all the attendees who has attended and who has make this session successful. And thanks to one and all. Thank you. And with this, I'm going to end this session. And thank you. Good night.
Thank you very much. Thank you.
Thank you very much.
Thank you. Bye-bye.
Thank you.
Thank you, sir.
Thank you.